first of all, I want to thank Dr. Rao for inviting me for such a wonderful platform. A lot of people here from different hues, and it has been like Fine Arts 101, and I'm adding more color to it. So the issue that I'm going to cover is, which has not been really covered as natural models here. So advances in the canine mammary tumor research as a natural model for human breast cancer. I won't go into the details because already a lot of details have been discussed about the breast cancer. But only thing which is little alarming is this one, that by 2030, there will be most of the cases of breast cancer are likely to be in the developing world like India. And many people have been discussing about the cities here. And in Punjab villages, in Sangrur district, in Bathinda district, there are a lot of cases of breast cancer. Probably unrelated, we don't know what is the etiology. I'll talk about that a bit. Well, before I go further, let's look at the comparative oncology. There has been a great surge in this branch over the last 50 years, and several models have been developed, including rat and mouse models, as we have discussed. But dog is a wonderful model, naturally occurring cases. The previous speaker had talked about that. So here, several tumors can be studied. And most important issue here has been looking for the new therapies. You have known that. So why dog as a model? That's the question. There are three major considerations when you look for a model in any cancer research, comparative oncology. The first issue is ethical consideration. The second is clinical consideration, and the third one is financial considerations. So if you look at the ethical considerations, you have to obtain the maximum information from the same number of animals and use the methods which cause minimum potential pain as well as distress to the animal. That should be the aim ethically. And if you look, dogs, as they suffer naturally or spontaneously with a high frequency of mammary cancer, therefore, they fit very well into these considerations rather than the need to induce, or the rats are made to suffer from tumors, which is not ethical. And the latest treatments that will be discovered that shall be applied to the dog first and before its extrapolation to the human mammary tumor, that puts in a positive position the dog. And clinical considerations, if you look, we have the first thing is that canine mammary cancer is a very common tumor, and it is more common than humans. The dogs suffer almost as double as the humans. Some breeds even suffer triple the incidence. And very important issue here, because the pets or the dogs are exposed to the same environment, habit and habitat is similar, so they develop the cancers in pretty similar manner as the humans. The dog's metabolism matches the humans better than the laboratory rodents. That would make the trials much more meaningful than the rat and mice. If you look at the size and weight of the dogs and cancer biology, that makes it easier than rat and mice for intratumoral monitoring, sequential biopsies, molecular diagnosis, imaging studies, and all others. If you want to take sera samples, blood samples multiple times, and even multiple biopsies. And also, the dogs receive special health care as humans. They can live with the owner up to a long stage into the old age, and it's easier to follow them up. Looking back into the financial considerations, there has been a study by a coalition of partners, including hospital managements, including the leadership issues, and including the pharmaceuticals, all of them, and which says that if we invest $1, it produces $3 worth gain in cases of the diabetes or cholesterol-lowering drugs. If you develop antiplatelet therapy, the gain would be $2 to $6, whereas additional dollars spent on hormonal therapies finding for the breast cancer, that would save you about $27 of health gain. So that means the preclinical trials can be much better be developed in dogs rather than the rodents. Well, summing up the process, the cancer in dog is spontaneous. It is, again, the second most common tumor after the skin tumors in the dog. And the median age is 11 years, 6 months, which is equivalent to 62 years. And we have been talking a lot about that age and advancing of the cancers in the human population in India to the 5th decade. So that, there is a short lifespan. So that gives us a positive thing. What we intend to do in humans in 60 years, or in terms of the reoccurrence or metastasis, if it takes, after treatment, the 5-year survival, here the survival would be 6 months to 1 year. That cuts short the mechanism for looking for the therapy. So in a shorter time of span, we would have more research and better outcome. Incidence increases with age, that we already said. The cells of origin are stem cells, CSCs. The metastasis is similar and it evolves rapidly. The similarities in prognostic factors. And one thing about genetics, it has been seen now that the dog genetics resembles, it is quite homologous with the human genetics, and about 85% genome of the dog is similar to man. And above all, habit and habitat. Well, before I go further, there are 5 pairs of the mammary glands in dog instead of the 2 mammary glands or 1 pair in the humans. And it has been seen that sometimes the whole chain is involved, and sometimes only 1 gland is involved. And it has also been seen that different mammary glands, they may have different grades of the tumor, different evolution of the tumor, and also sometimes you find that tumor in this gland could be different than this. This could be a simple carcinoma, this could be a complex carcinoma or carcinosarcoma. So that means we have an added advantage here of looking at the dog as a model in cancerogenesis, breast cancerogenesis. And you have seen the lymph nodes also. There are 2 sets of lymph nodes here rather than 1 set, and that makes it also better. And this is just one study which shows that it increases with age and the maximum, and this is our own study, and that's in humans. And those are the gross lesions, radically similar to the humans. And location of the neoplasm is maximum in the last pair, in the posterior pair. So why? Because the size of this mammary gland is the biggest. Probably the estrogen receptors or the hormonal receptors are more there, and that's why there's likely a chance of developing the tumor more. Looking back into the etiology, we have already been looking at for several days now, 2.5 days almost. So hormones and growth factors, receptors, food. We have talked about obesity right now, amplification of HER2, suppressor genes, many of them. In fact, it is only a few which are mentioned here. And that's the development of how the tumor would develop, and a lot of, we have been talking about the caretaker pathway, the gatekeeper pathway, the driving genes, and the associated genes, all that. And it has also been seen that if you look at the undifferentiated mammary cancer and the differentiated, the receptor expression is much more here, and that's what looks for the therapies we have been talking about. And cytology is radically similar between the two, and that's the cytology. But here, another thing is that we have several tumors, about 40% tumors, which are mixed mammary tumors. So they show a differentiation of the cartilage, they show a differentiation of the bone, which usually occurs in the human salivary tumors. So that could be a better way of looking at the desmoplasia or the EMT studies. And that's a lymph node metastasis, lung metastasis there. And that's a bone marrow metastasis. That's a bone marrow. These are our own studies, bone marrow cytology showing the metastasis, micrometastasis. Histopathology is radically similar, so much so that the people believe that it's hard to differentiate whether the slide is from a man or from a dog mammary tumor. So that's what is shown here, the human and the canine normal gland, and that's ductal carcinoma in situ in both, radically resemble each other. Immunohistochemistry is pretty similar. The same type of modalities or molecular classification is used in dogs as in humans. Before I go into some more slides, I will be talking about the stem cells. We have been looking at that. So we have the cancer stem cells, which are capable of self-renewal, and they can become either luminal restricted progenitor or committed stem cells or basal restricted progenitor cells, or there can be bipotent progenitor cells, means which are capable of showing both luminal and basal characteristics. That's very interesting. And then they differentiate into the luminal or alveolar type or ductular type here, both from this pathway also, and also they can differentiate into myoepithelial cells. So based upon the human classification, we did try to do some classification of the canine mammary tumors. The same scheme followed. And this is the ER positive receptors. And this is the HER2 positivity. And based upon the same classification, we have been able to analyze the tumors, and there are some, as in humans, which remain unclassified, and basal is more complex, basal oblique luminal. And luminal A, B, HER2 overexpressing is okay, as we have been talking all the days through. But if we add more markers into this, if we have an extended surrogate panel of antibodies, the situation becomes more complex. So this is P53. This is smooth muscle actin and P63. So we added several more markers, and then we tried to see the classification changes a lot. So as I told you earlier, that there are some committed stem cells which can either go basal or luminal, have characteristics of both. So that became, these became the major, basoluminal A and basoluminal B, which in fact, the difficulties in classification already have been pointed out by most of the people. Another study which we did, I'll be just giving you an overview because time is very short. I'll try to finish in time. So epithelial mesenchyme transition in mammary tumor, that was another issue which we tried to study. So as we know that this has been discussed at depth by people, but still I'll try to do some basic work on that. So we have epithelial cells. All individuals start as epithelial cells. And then the mesenchymal cells arise. And majority of the tumors, human and canine, they are of epithelial origin. And that's what we have been talking, the previous speakers, Dr. Mehta has told in detail about the progression and how the EMT could be responsible for invasion and metastasis. And that's in brief, looking at the epithelial and mesenchymal, how the cells which remain together cohesively, they become detached from the primary site, behave as penetrate through the connective tissue barriers, and then enter the circulation and then reinvest into the other tissues as metastasis. The first evidences, or most of the evidences so far of EMT have come from the in vitro models, as people have been showing slides here. And this is a mouse, transgenic mouse model. And you see here the green and red contrasting. So vimentin here is green, and the cytokeratin is red. And this is our own study. So when this was about 8-9 years back, this was the tumor in Achi, when I tried to stain that with dual staining for vimentin and cytokeratin, you can see that red vimentin and black cytokeratin. So how the tumor which looks like an epithelial tumor shows that vimentin? That's the question. So we have been talking about the progressive loss of cytokeratins. So that's another thing. As the tumor progresses, from the anaplasia progresses, you see there is a loss of cytokeratin. And also there is a progressive loss of the adhesive molecules, that's E-cadherin. So you can see here a lot of expression of E-cadherin. There is a reduction and there's a marked loss as it progresses in grading. And there is also a progressive gain of vimentin expression as the tumor progresses. See, from here to here. Instead of E-cadherin, the N-cadherin is gained. That's a immunohistochemistry showing the N-cadherin there. There is also production of matrix metalloproteinases, which helps the tumor cells to penetrate through the ECM. We have also been talking about the stemness and the cancer, and these are some stem cell markers. We also did some relative quantification PCR, and which showed that there is a relationship between the HER2 and the EMT. Then we also looked by the SHIS, the chromogenic in situ hybridization. And again, you can see that expression there. I'll skip this. In fact, snail, slug, twist, several others are the propagators of EMT, which has already been discussed here. And there are several phenotypes of EMT which have been classified, type 1, type 2, and type 3. And the EMT has now been related to stemness, which my previous speakers have discussed, drug resistance, inflammation. Another study that we did, it's a dual staining. We were looking for lymph angiogenesis. We did several markers like LIVE1, PROX1, podoplanin, VGFR3. So this is a dual staining where you can see both the things together. So these are the lymphatics, the black colored, thin walled, and this is neovascularization or neoangiogenesis. So the inference, I'll go to the next because time is already over. So possible role of pesticide residues in canine mammary tumor, that was another issue we tried to cover down the lane over 10 years now. So we did that gas chromatographic analysis on several insecticides, and they were present in the tumor tissue as well as in the plasma or serum. And also there was upregulation of aromatase. So the inference drawn from this study was that insecticides like dialdrin, heptachlor, alpha, gamma, HCH, SCH may be related to oncogenesis, possibly by converting the androgens to estrogen pathway. So the overall, so what I tried to show you in brief, a little bit of what we had done, and I tried to propagate the naturally occurring model, the stress on which has been laid by several previous speakers, and Dr. Mishra, who was talking about the obesity, he was talking about lack of these models. So overall, if we look, the mammary gland and their tumors in canine and humans are pretty similar. Spontaneous tumors in dogs is a good model, naturally occurring over a short time of period, saving a lot of money for studying both the genetic and the epigenetic or the environmental factors, as I have already shown. Can be used for developing new diagnostic, prognostic, and predictive markers. And for this purpose, the pet population is greatly underutilized, and it continues to live with the owner that is saving a lot of money. And you get a lot of information which you cannot get in the rat and mouse model because you don't know what is the behavioral change, how it is going on, and what is developing, and you have to look for everything. Here, the owner can tell you many things. Another point I will bring to your fore is, can dogs be predictors of human malignancy rates and etiologies? Because they live in the same environment. So looking at a dog population in a particular area, and that could be a future propagation. Another thing, this dog as a model fits well into the one health, one word, one medicine concept. So thank you very much.